Do not forget, you can support the channel with a like, and you can also subscribe to be aware of the latest published videos. Thank you. We hope that here you can find the latest news, ideas, and discoveries from the scientific world.
Stanford scientists identified genes linked to familial brain cancer. A global collaboration directed by a researcher from Stanford Medicine has discovered over 50 genes associated with glioma, a rare brain cancer. While the majority of gliomas occur spontaneously, a small fraction is inherited. In 2013, Carrie Davis Lebovich and Hadley Rierson, two sisters, found themselves grappling with frustration. Their father, John Davis, aged 69, had recently received a glioblastoma diagnosis, a rare type of brain cancer, the same one that had taken their grandmother's life 3 decades before. Given the rarity of glioma, with only roughly 24,000 diagnoses in the United States annually, they believed the odds of lightning striking twice in the same family were slim. Our first question to his doctors was, "Is this related to the glioblastoma that killed our grandmother?" Rierson recalled. And they all said no. Brain tumors happen randomly, the sisters were told. Their family's experience was just a coincidence. The sisters didn't believe them. After some dogged research, Lebovich and Rierson found Melissa Bundy, Ph.D., who was then at Baylor College of Medicine in Houston. Bundy, now the chair of the Department of Epidemiology and Population Health at the Stanford School of Medicine, directs an international consortium called GliaGene aimed at identifying genes involved in familial glioma, a class of brain cancer that includes glioblastoma. Bundy assured the sisters that, yes, despite what their father's doctors had told them, a small minority of glioma cases are familial. Pinpointing the genes involved could not only help identify which members of affected families have an increased risk of brain cancer but also shed light on the biology of the disease and drive future treatments. To do so, they needed genetic samples from as many patients and their family members as possible. The sisters immediately signed on. Now, Bundy, who is the Associate Director for Population Health Sciences at the Stanford Cancer Institute, and her GliaGene collaborators have identified several genes associated with familial glioma, 2 of which are also associated with ovarian and colon cancers. They also found mutations in 3 locations of the genome called non-coding regions that affect which genes are made into proteins. They described their findings in an article published April 28 in Science Advances. The identification of these new genes and non-coding regions is of immense value to families affected by glioma, Bundy said. The discovery provides the opportunity to explain to affected families why they are at risk, offer peace of mind to those who do not carry the causative mutation, and improve monitoring for those who do. Other senior authors of the study are Matthew Bainbridge, Ph.D., Associate Director of Clinical Genomics Research at Rady Children's Hospital San Diego, and Benjamin Deneen, Ph.D., Professor at the Baylor College of Medicine. Dong-Yo Choi, Ph.D., a postdoctoral scholar at Baylor, is the lead author of the research. Rarest cases within a rare cancer. Gliomas encompass several subtypes of brain cancers including glioblastomas, astrocytomas, and brainstem gliomas. They arise from cells in the brain called glial cells that support the brain's neurons. Although some are slow-growing and relatively treatable, the prognosis for many of these cancers is poor. According to the National Brain Tumor Society, the average survival of glioblastoma patients is 8 months after diagnosis, only 6.8% are alive after 5 years. Most gliomas are sporadic and seem to have no clear genetic cause. Only about 5% of gliomas are familial, afflicting 2 or more members of the same family. Bundy and her colleagues want to identify the genes involved in these, the rarest cases of an already rare cancer. But to do so they need to enroll as many people as possible in the GliaGene study. So far, the study has enrolled over 15,000 people and identified about 350 cases of familial glioma. John Davis died in July 2014, just 13 months after his glioblastoma diagnosis. In December 2014, Bundy and other members of the GlioGene Consortium announced the discovery of one of the first genes associated with familial glioma, POT1. Mutations in POT1 carry an increased risk of developing glioma, the researchers found. To find additional genes and DNA regions associated with brain cancer, Bundy and her colleagues sequenced the entire genomes of 325 people with glioma from 304 families with a history of the disease. They compared their genetic sequences with those of more than 1,000 controls without brain cancer. They found 6 mutations in one gene, called HERC2, that were associated with familial glioma. The protein made from the HERC2 gene is involved in the repair of damaged DNA and the control of the cell cycle. Although these roles are shared with many other cancer-associated proteins, the HERC2 protein was not previously associated with cancer. 2 other genes, BRIP1 and POL, that were also mutated in familial glioma cases have been associated with ovarian and colorectal cancers, respectively. The researchers used CRISPR genetic engineering to delete several candidate genes in embryonic mice treated to develop glioma and saw that the loss of 3 of them, DMBT1, HP1BP3, and ZC3H7B, correlated with a decrease in survival and an increase in tumor growth in the animals. All told, the researchers identified 54 mutations in 28 genes or non-coding regions that were associated with familial glioma in 50 out of 304 families in the GlioGene study. Many of the genes are involved in cell division, blood vessel development, and immune regulation, all factors that can contribute to tumor growth. This is such a rare disease, Bundy said. The worst part is there is currently no effective treatment for many brain tumors. My hope is that one day I can answer people who ask me what is my chance of developing a glioma? Such a huge relief. For Lebovich and Rierson, the study brings hope. Any lingering chance that their family's brain cancers were simply coincidental vanished when their father's sister died from a glioma in 2017. They've devoted their time and energy to spreading the word about familial glioma and encouraging people in affected families to join the GlioGene study. It couldn't be easier or less painful to participate, said Rierson, noting that participants simply submit a sample of saliva through the mail. Hadley and I had so many doors shut on us, Lebovich said. It felt very lonely. It was hard to get information, we were told to move on. But when we reached out to Melissa, she was on the phone with us that same day. It was such a huge relief. She has been an unbelievable partner. Melissa and the GlioGene collaborators are our only hope for the future generations of our family, Lebovich continued. But we need people to participate in the study. The more genes we know are associated, the better you can screen potential carriers and possibly tailor treatments. But you can't do anything if you don't know the genes.